So my name is Mingyang Lu. I'd like to thank the organizers to give me the opportunity to present. So for my lab, we're interested in to develop computational methods and apply them to understand the gene regulations of cell state transitions, such as cell differentiation. So our strategy is that we try to integrate the literature-based gene regulatory network informations and omics data, and then apply bioinformatics methods to construct gene regulatory networks. And then we want to integrate systems biology modeling afterwards. So we want to do simulations and do modeling and try to identify the core regulatory circuits. And then we can use simulations to figure out how the circuits works, and also if we need, we can do some refinements to make the model better. So essentially, we're trying to integrate the top-down and the bottom-up systems biology together. Over the last few years, we are trying to develop computational methods along these two different approaches, including developing methods for ensemble-based ODE simulations and modeling and network cross-training, and also circuits motif analysis based on the simulation results. And at the same time, we also want to integrate omics data. We integrate single-cell sequencing data, ATAC data, and also literature-based target relationship. So in today's topic, we are interested in to develop a computational methods to model gene regulatory networks using single-cell sequencing data. Especially, we're interested in a single-cell seq because it can delineate the cell state transition very nicely, because the gene expression usually is pretty continuous. As you can see in this example, they can clearly visualize the hierarchical structures of multiple step or bifurcations from the data. And there were methods to take advantage of single-cell sequencing data to characterize the cell state transitions, for example, by integrating time series measurements, pseudo-time inference, lineage tracing, and also RNA velocities. However, when it comes to network constructions, and although there were a lot of exciting methods out there, as we can learn from the previous talkers throughout a few days in the conference, but still the inference still had very low accuracies, as demonstrated in several benchmark studies, including the one I put it over here. Partially because the single-cell seq data are pretty noisy, with a lot of job artifacts and also biological noise. And it's hard to infer the causality from these associations. And another important aspect is, to me, many studies currently focus on accurate mappings of gene regulatory interactions, but it's a lack of evaluation of GRNs to see whether they actually form a function as a working systems to drive cell state transitions. So that's something we think there's an opportunity for us to tackle these problems. So we take advantage of the hierarchical structures that there are about thousands of transcription factors, mainly target genes, basically regulated by them, and the interplay between the transcription factors were allowed to drive the state transitions. So we want to take advantage of these informations to help us to infer the core transcription factor networks. And then based on the networks, we can use to see whether this can be the first principle for us to model the cell state transition patterns that we observe experimentally or not. And with that, we can help us to refine the model. So this project is done by my graduate student, Yukai Yu from BioE. So our goal is to develop computational methods to build dynamical models of GRNs directly from single-cell sequencing data, particularly time series data, and together with the literature-based target relationship. As I mentioned earlier, this is a typical approach we do to integrate bioinformatics and mathematical modeling together. And it's based on the temporal gene expression patterns because they capture the cell state transitions. And then we do parameter optimization based on the nonlinear ODE models typically used in systems biology. So we develop methods on the contents of particular public available time series single-cell sequencing data for iPSC differentiation towards the mesendoderm and to definite endoderm. So based on the projection of the data to pre-CA, you can clearly see the sequential transitions through multiple states here. So that's what we are trying to model here. So the method is called network modeling based on dynamic equation simulations on NetDAS. So we first do pseudo-time inference. It's pretty straightforward in this case here. We can do principal curve. And then we align the gene expression for every genes along the pseudo-time. As you expected, it's pretty noisy. And we apply a method called pseudo-time DE that's developed by Professor Jessica Lee's lab. And we like it a lot so that I can actually smooth the gene expression to get a very cleaner data. The method is based on the Bayesian inference by assuming that the gene expression for every gene for every cell is a negative binomial distribution, where the mean gene expression along the skew-time can be fitted with a smooth spline curve. So therefore, we can get a very smooth gene expression for every gene, and then we cluster them together. In fact, we have to do our own ways to have a better results. But then we assume that if these genes in a group have a very similar gene expression because they're likely to be regulated by a core transcription factors. And therefore, we can do some gene set enrichment analysis based on the TF-target relations to identify the enriched transcription factors from that. So we utilize the TF-target databases compiled from literature that we published previously, the NetDAM methods. And also we use popular methods, the RCS target from scenic for that purpose. So basically, then we can construct an initial network where genes transcription factor regulating each other. So certainly, we can also apply some new methods that developed recently and to get the initial network as well. And also we can incorporate some additional data sets like ATAC-seq or other data sets. So then we utilize an optimization approach to establish an ODE-based model. And meanwhile, we will be able to optimize the network. To zoom in a particular step at the end. So basically here in an example where SNAIL1 is a transcription factor that regulated by another 4 transcription factors. We already know their gene expression time trajectories from before. And then we fit an ODE model as shown here. So this is a very typical ODE that we use in systems biology. We have the production and linear degradation, and also the products of each from transcription factors, which is basically a typical Hill kinetics that we describe. And I particularly want to emphasize one parameter lambda here, which represents the maximum fold change for every regulation. So this can tell us whether an interaction is strong or weak, and also the nature of activation inhibition nature as well. And when we do the fitting by using the time trajectories here, and then we also, in order to make the optimization robust, we have a regulation terms so that we eliminate those interactions that's not real, that's not relevant. And meanwhile, we do 5-fold cross validations, and we also store from multiple initial conditions parameters in order to have a robust fitting. So here's an example of the testing. So basically, we have some random gene expression profile trajectories for regulators, and then simulate the target gene expressions. We particularly put 2 gene regulators, 1 and 3 here, as a decoys, because the lambda is essentially 1, there's no interactions. And the methods successfully eliminate these 2 interactions. And for the other 2 interactions, although the lambda values is not exactly the same, but we get the right activation inhibition nature for interactions. And here's another example for that, where we can identify 2 decoys from 5 interactions. So we then go ahead to do a systematic benchmarking by generating randomly pick some regulators and decoys and simulate them and compare with other methods, including methods that take the time series data. And we found that that indeed has advantages to identify the decoys out of the real interactions. And even when the gene expression noise, there's gene expression noise in the data, in the time series data. And then we can go ahead to take every transcription factors and identify their regulators and the natures of the regulations. And here, for each individual target genes, we find the fitting is pretty well, except one genes that one here, where the reason why it's a little bit off because there was only 1 regulation here. So the fitting didn't go very well with that case. But anyway, so we now have the optimized network. It's actually pretty tough to make the comparison with the ground truth because we usually don't know the ground truth. And in our case, because every interactions in the initial network already have some evidences because they're either coming from a TF target database from the literature, or there's some TF binding motif evidences from that. But still, we go ahead to literally check manually all the interactions from the initial networks and to figure out whether there's direct experimental evidences for those. Then we can make some statistical evaluations of performance of the network. Although there were some caveats here, because even from categories like false negative, which means that there's no interactions, but we limited them. But the elimination probably because this is content specific for definite endoderm differentiation. And also there's an unknown category where we identify some interactions we cannot find the evidence from literature. That could be new discoveries. So it's a little bit caveat for this comparison, but we compare with a few other methods and we are among the top of the methods to infer the networks. But the gain for our methods is also we have a GRN model, ODE models that we can do simulations on. So interestingly, for the networks, we identify some stemness transcription factors, their activity is going downwards, and some EMT transcription factors, the activity is going upwards. And we're suggesting there's a coupling between the stemness and the EMT. And we indeed found evidences about that for these contents. And the most exciting part of this story is that now we have an ODE model, we can then do simulations to drive the cell state transitions. And we know experimentally that these cell state transitions, cell differentiation was achieved by a cocktail of growth factors with the targeting the OCT4, SOX2, and etc. But it's not very well characterized. But we then just go ahead to perturb each transcription factors or a combination of two transcription factors according to their gene expression, observed gene expression dynamics, and simulate them and compare and see how the driving when nodes will be able to drive the gene expression dynamics for other genes, and then we compare them. So we found that by driving one gene, usually they didn't do a very good job, and yeah, I don't know why. I only see one. Actually, there were multiple curves on that, so you just should believe me that by driving one gene, it didn't work very well. But driving two genes, we actually can see very close observations of simulation results and the data. And interestingly, by driving one gene, we also find some hysteresis, which usually occurs in the non-dynamical systems. But if you do linear model, usually you will not be able to observe them. So suggesting by driving one gene, there's a barrier during cell state transitions. And we see evidences about the driver genes as well. And yeah, sorry, I don't know why it's just not working. Okay, yeah. So we also start to do additional simulations to check how the network will be able to robustly capture the cell state transitions. We do additional recipe simulations, the methods we develop to simulate ensemble models where we use the network topology as the inputs and then generate ensemble models of random kinetic parameters to see whether the simulated gene expression patterns can be captured from single scRNA-seq data, how consistent they are. We do found for NetAds, we will be able to fund simulation. We only use the topology as the inputs, no parameters at all. And we can still see the shape of the cell state transitions very similar to the experimental data, but for other methods, we'll not be able to see them. So we can further, the network is still pretty complex. We can further just group the genes based on their gene expression dynamics and then lump them together into one node. It's like cross-graining, and which is mentioned in the previous talk. So we will be able to apply a method we previously developed and demonstrate that we can cross-grain the network into 5 nodes where the gene expression, simulated gene expression pattern looks very similar to each other, and they pretty much capture the state transition patterns that we observed in the data. And we're still it's still ongoing studies, and we try to understand what the network means. And we do observe the coupled toggle switch circuits and also some feed-forward loops, what probably will help to establish the sequential transitions during the cell differentiation process. So in conclusion, we develop conditional methods NetAds to build dynamical models of GNNs using single cell sequencing data. This is based on nonlinear ODE-based model. We optimize it based on time trajectories, and we'll be able to find the driver genes based on the gene expression simulations. And compared to other methods, NetAds has the advantage of capturing cell states and also the transitions, the orders of state transitions. Thank you, and I'd like to take any questions.
Do we have any question for our speaker? Yeah.
Hi, is this on? Yeah. Hi, this is a really cool talk. Thank you for sharing all the work with us. I was just wondering, is the software available if we want to test it out? And also, do your networks include known key factors for endoderm development like SOX17 or FOXH1? Thank you.
Yeah, so for the software, we're still putting them together. So that will be available very soon. So once we have the bioCAD papers established, and we will publish the GitHub. And in terms of some real transcription factors, that's indeed some of them it was not included in our TF target databases, especially those from the literature. So, but for some key factors, we can add them. We can also integrate other methods to incorporate other transcription factors.
Thank you. Yeah, please.
I was wondering if you've thought about incorporating sequence-based TF target prediction model, because that will give you much more associations within your GRN.
Yeah, that's a good point. Yeah, we're thinking about that. There are methods by integrating ataxia data, by using sequencing. So I really like those approaches. So that's the next step. We want to integrate ataxic data, multi-omics data. And we also want to study the cases where state transition is more complex, like bifurcations or reprogramming.